In this lecture, we'll be talking about protein translational modification. So why are proteins modified? Well, you can regulate activity. A modification can turn an activity on or turn it off, or it can generate a different function. Also, for protein interactions, modification site can be a binding interface. Subcellular localization, modification site may be a targeting signal or a membrane anchor, and then aging modification can identify proteins for degradation or target a protein to be scattered. Here's a prokaryotic cell. In prokaryotes, you have your cell wall and then plasma membrane, and then inside, all within the internal cytoplasm, you have your nuclear DNA, you have your plasmids, you have your ribosomes, and you have your transcription and translation occurring in the same compartment. This is our contrast with a eukaryotic cell as pictured here. Eukaryotic cells have an external membrane, but also have these membrane-bound organelles, and the nucleus is separated out from the rest of the environment. And so you have things like the mitochondria. You have, in addition to your nucleus, you have your rough and smooth endoplasmic reticulum. You have Golgi apparatus. And so these membrane-bound organelles are important for post-translational modification. If we look at the transitional elements of the endoplasmic reticulum, we have the cis Golgi turning into trans, and then you have budding off to form lysozymes, secretory granules, as well as continuous or constitutive secretion. Now, in all these processes, you have membranes by which you can have post-translational modifications occurring. By definition, post-translational modifications are covalent modifications that take place either during or after the assembly of a polypeptide chain. There are two types of PTMs. One is cleavage or addition of peptide bonds, and then the other is the derivatization of amino acids. So in terms of the types of modifications that we're going to be looking at, we're going to look at the first class, which are proteolytic cleavage, and then the rest, which are modifications that are made on the amino acid: glycosylation, phosphorylation, acetylation, methylation, sulfation, addition of lipids. These are two that we'll be focused on: Myristoylation, Farnesylation, Hydroxylation, Ubiquitination, and then NNC terminal mods. Like I said, within the eukaryotic cell, you have cellular sites of major post-translational modifications, starting from the cytoplasm all the way down to these different mitochondria, endoplasmic reticulum, Golgi apparatus, secretory vesicles, and granules. And depending on what the site is, you have different modifications. So, for example, in terms of cytoplasm, you have the removal of initiating methionine, acetylation of the alpha NH2, myristoylation of the alpha NH2, O-glycosylation with GLICNAC, addition of palmitoyl groups, virus polyprotein processing. In the mitochondria or chloroplast, you have cleavage of signal peptide, endoplasmic reticulum, again cleavage of signal peptide, core glycosylation of asparagine residues, addition of palmitoyl oil, glycosyl phosphatidylinositol groups, carboxylation of glutamic residues, hydroxylation of procollagen, pro and lyse residues, disulfide bond. In Golgi apparatus, you have my modification of N-glycosyl groups, O-glycosylations with GLICNAC, sulfation of tyrosine, and amidation of the alpha carboxy group, proteolytic processing occurring in the secretory vesicles and granules. Ribosomes engage the endoplasmic reticulum membrane through interactions of the signal recognition particle, or SRP, in the ribosome with the SRP receptor on the endoplasmic reticulum membrane, shown here. So proteins that are membrane-bound or are destined for secretion are synthesized by ribosomes associated with the membrane of the endoplasmic reticulum, and what happens is, as the ribosome is generating or translating the mRNA, it generates the signal peptide, which gets recognized by the SRP, which then allows the ribosome to then bind to the ER membrane and lets the protein come and translocate. And as it's being processed through, it gets cleaved by a signal peptide, and then it gets secreted through. The important part of this is this signal peptide, and the signal peptide is usually 13 to 36 predominantly hydrophobic residues, which then recognize this SRP receptor. Proteins that contain this signal, right, before it's cleaved are called preproteins, and we'll talk about definitions in the next slide. So there are proteolytic enzyme definitions. Preproteins are proteins that contain that signal peptide. It's in the N-terminus. Proproteins are protein precursors that are biologically inactive until further proteolysis. Preproteins are proteins that are destined for secretion, are also further proteolyzed following secretion, and therefore contain prosequences. Enzymogens, which are the same as proproteins, they're proteolytic proteins that are inactive until proteolysis. Cleavage sites are determined by the identity of the amino acids, specificity of protease, and folded conformations of zymogens. So protein sequences that bear these signal peptides undergo proteolytic processing. So these zymogens are full length here, and then as it's being undergoing development, it then gets processed. And so in the anterior pituitary, this gets processed into these two peptides, which then gets further processed in the intermediate pituitary into these four. So why is this common for secreted enzymes? Some peptides, like enkephalins, are too short by themselves, and so just generating a short amino acid sequence can't be done. And so this, by processing it down to like shorter peptides, you're able to generate them. And then they prevent premature activation of hydrolytic enzymes. In eukaryotes, the glycosylation or attachment of sugars are prevalent. The major form of protein modifications in eukaryotes are secreted and membrane proteins. Sugars are added in the endoplasmic reticulum and Golgi, and most proteins formed in endoplasmic reticulum are glycoproteins. In prokaryotes, there is no such thing as glycosylation, and so in eukaryotic organisms, many different forms and functions come about because of glycosylation, such as bioactivity, improved solubility, lengthening the half-life, and interacting with other cells. There are three important properties of glycosylation. One is the variable structure, which permits specificity in interactions with other molecules. Two is that it's hydrophilic, so it then stabilizes proteins in the aqueous solution. And three, it provides bulk properties, which affects the surface properties of proteins when the sugar is attached. Glycoproteins are oligosaccharides covalently attached to proteins, and glycolipids are oligosaccharides covalently attached to lipids. The predominant sugars found in glycoproteins are glucose, galactose, mannose, etc., shown here. On the next slide, I'll show you the different structures. Carbohydrates are linked to proteins through either O-glycosidic bonds, shown here, or N-glycosidic bonds, which are shown here. In N-glycosidic linkage, the linkage is through asparagine as an amide bond. In O-glycosidic linkages, it's a linkage through the hydroxy group of serine, threonine, or hydroxylysine. N-glycosylation occurs co-translationally when the N-terminus emerges into the ER, or endoplasmic reticulum. O-glycosylation occurs in the Golgi as post-translational modifications on already folded protein. Here are the sugar monomers. Like I said, glucose, fructose, galactose, mannose, xylose, sialic acid or nana, and acetylgalactosamine, which is glycanac, and N-acetylglucosamine or glycanac. So it's my expectation that for exams and tests, you are able to know the structures of the sugar monomers for the post-translational modifications. Also, O-link sugars, the synthesis of O-link glycoproteins happens stepwise by the addition of nucleotide-activated sugars directly onto the polypeptide. So here are some nucleoactivated sugars, UDP, GDP, CMP. The attachment of sugars is catalyzed by specific glycoprotein glycosyl transferase, and evidence indicates that each specific type of carbohydrate linkage in the O-linked glycoprotein is a result of different glycosyl transferases. N-linked sugar. The three major classes of N-linked carbohydrate modifications are high mannose, hybrid, and complex. We'll talk about their structures in the next slide. And the major distinguishing feature of the complex class is the presence of sialic acid, whereas hybrid contains none. The site of carbohydrate attachment to N-linked glycoproteins is found within a consensus sequence of amino acid. It's asparagine, X, and then serine, threonine, or cysteine, where X is any amino acid except proline. In N-linked glycoproteins, they all contain a carbon core of carbohydrates. This core consists of 3 mannose and 2 glycanacs. So all of them, the complex, hybrid, and a high mannose, have that. And then they're distinguished by what's on the outer core. So high mannose over here has only mannose, which is easily observed. Hybrid have various sugars and amino sugars, so it has some mannose and other sugars. And then complex are similar to the hybrid type, but then in addition, they have these sialic acids, which hybrids do not have. So most proteins that are secreted or bound to the plasma membrane are modified by such carbohydrate attachments. The part that is modified in the plasma membrane-bound protein is the extracellular portion of the protein that is modified. There are intracellular proteins, and those are less frequently modified by carbohydrates, but there are carbohydrate attachments that confer functional activities on these proteins, much of them having to be involved in transcription. In initial glycosylation in the endoplasmic reticulum, you have this membrane-bound precursor oligosaccharides that's formed on this dolicolipid. This is then transferred onto asparagine as the polypeptide is being generated, and then this transfer is complete on the endoplasmic reticulum lumen side. So processing in the Golgi is shown here, where it's going from the endoplasmic reticulum lumen to the Golgi lumen. So after the attachment of that core glycan from the previous slide of a protein in the endoplasmic reticulum, the glycan becomes modified as it passes through. So types of processings depend on the identification of protein, the cell type, as well as the physiological state of the cell. And as you can see, as it's going through the ER lumen, you have different enzymes clipping off the core, and then you have, as it starts to pass through the Golgi lumen, you have again more trimming occurring, and then additions happening as you go through this whole process, and then you get ultimately towards the end a complex oligosaccharides that is very different from what you had started off with here. And depending on the location of the ER or the Golgi lumen, you can then identify the protein's state or where it has traveled, because it will have different modifications. Glycosylation can also help in protein folding. So if you have a completely unfolded protein, you have glucose trimming, and that trimming starts to help it to fold, which then helps it get recognized on the ER lumen, which then also helps it get processed so that it's in the appropriate conformation so that it becomes normally folded. If it's not, it's incompletely folded, you have glucosyl transferase coming in, bringing it back to the cycle until it gets completely folded. Proteoglycans are also important in the extracellular matrix. Proteoglycans are protein backbones to which one or more glycosaminoglycan chains are covalently attached. They're secreted and have end-linked oliogsaccharides attached to other side chains. They're made of core proteins and many polysaccharides change, and the core proteins vary in molecular weight from 11 to 222 kilodaltons. And the number of glycosaminoglycan chains go from 1 to 100. They form hydrated gel and they're as part of the extracellular matrix. They resist compressive forces, regulate traffic. They can regulate secreted protein activity, for example, chemokines and inflammatory response. And most CCM proteins and growth factors have these binding sites, as you can see, for GAGs or glycosaminoglycans. GAGs are polymers of disaccharide building blocks, as you can see, shown here. The sugar backbone can be sulfated at different positions, and depending on where it's sulfated, you can generate various patterns. And so a simple hexasaccharide can have over 4,000 different sulfation sequences. There are 4 types of glycosaminoglycan repeating disaccharides: heparin sulfate, shown here, chondroitin sulfate, keratin sulfate, and hyaluronan. This is unsulfated, shown here. Biosynthetic enzymes modify specific positions within the sulfate, epimerize some glyc-A to ido-A, and in KS, meaning keratin sulfate, may add monosaccharides, shown here. This is the structure shown here below. In general, glycosaminoglycans are sulfated and usually attach to protein backbone through xylose linked to serines. And they can also be attached to serines through N-glucosamino and can be also N-linked. The signal for attachment of chondroitin sulfate is preceded by 2 to 3 acidic amino acids, and then it has this signal SGXG, where X is any amino acid. So the different glycosaminoglycan families can be involved in different developments or biological events. So here's chondroitin sulfate, which is important in brain development, learning, and memory, and cancer. Heparin sulfate, brain development, blood clotting, and cancer. And keratin sulfate, macular corneal dystrophy. Membrane proteins, or GPI, anchors, or glycosyl phosphatidylinositol, or GPI anchors, are these lipid attached sequences that can then attack and form a covalent bond to proteins to then make them membrane attached. Trypanosomes actually can shed these proteins to avoid immune attack. Phosphorylation is another type of post-translational modification. This is in fact the most common post-translational modification for proteins in eukaryotes. Enzymes can be turned on or off, and the energy is derived from ATP. So ATP plus protein will generate your phosphoprotein and ADP. In general, the residues that get phosphorylated are serine, threonine, and tyrosine, and here are the phosphorylated versions. So the kinase will, for example, recognize serine in the presence of ATP, generate the phosphoprotein, and then protein phosphatase does the reverse reaction by removing it. And so you're essentially taking something that has a hydroxy group and then adding negative charge to it by adding this negatively charged phosphate group into the protein. Which amino acid is phosphorylated depends on the primary sequence around the amino acid, the accessibility to kinase, as well as the specificity to kinases. Again, and kinases either can turn the protein off and phosphatase turn the protein on and phosphatase turn it off, or vice versa, as shown here. Phosphorylation as molecular switches. So the molecular switches fall into 2 main classes that operate in different ways. although in both cases it is to the gain or loss of phosphate groups that determines whether the protein is active or inactive. The largest class consists of proteins that are activated or inactivated by phosphorylation. So for these proteins, the switch is thrown in one direction by a protein kinase, which adds one or more phosphate groups to the signaling protein, and in the other direction by a protein phosphatase, which removes the phosphate group. So the other main so this would be in the case of ATP, which we've shown. You have ATP in the presence of that kinase. Add the phosphorylation and then signal out. Dephosphorylation with a phosphatase, and then it turns it off. The other main class of molecular switch involves GTP binding proteins, shown here. In this case, these switches are between an active state when GTP is bound. So when GTP is bound, so there's no hydrolysis occurring, just phosphate being added. No, what's happening here is you have the whole GTP being bound, and that turns it on. When the phosphate gets released, and it's GDP, turns it off. Phosphorylation can also impact motor proteins. It allows movement of chromosomes during mitosis, organelles along molecular tracks, as well as moving enzymes along DNA during DNA synthesis. So as an example, you can have ATP binding to the protein in one conformation. So once it's bound, here's your it takes conformation 1 to conformation 2. Once ATP is hydrolyzed into ADP and phosphate, you go from conformation 2 to conformation 3. And so now you have one leg down, then both legs down, and then this leg moving in. And then when it releases, you go back to the state of 1. And it's irreversible and going into one direction. Acetylation is another type of post-translational modification, but it now exclusively looks at lysine. So in the presence of lysine, acetyl coenzyme A, and histone acetyltransferase, it'll add on the acetate from acetyl coenzyme A onto the lysine residue. This will then bring a positively charged to a neutral amino acid, and you can have the reverse reaction happening with histone deacetylase proteins. So in addition to arginine methylation, you can also have lysine methylation. In case of lysine methylation, use the enzyme histone methyltransferase, again using SAMs or ADOMET, which then transfer that methane on top of the lysine. And so this can undergo not only mono but di and trimethylation with each addition of SAMs. And so what the methylation does is that once it's methylated, it takes something that's positively charged but makes it electron donating so that it stays more positively charged, so that it binds then tightly to the DNA, repressing it or silencing the genes. So if we look at methylation versus acetylation, in the case of acetylation, you're taking a positive charge in the acetyl coenzyme A and then neutralizing it. This then leads to histone activation. HDAX makes it positive again and then brings it back to its repressed state. And using HMT or histone methyltransferase, in the presence of SAM, you're taking this and then making it more positive and then more repressed, and then the opposite would be a demethylase, in which this would bring it back to the positive charge. Tyrosine sulfation is a post-translational modification mediated by one of two Golgi isoenzymes called tyrosyl protein sulfotransferases, right? 1 and 2. The the tyrosine protein sulfotransferases require the phosphoadenosine phosphosulfate, or PAPS, and that provides the sulfate group to transfer on to the tyrosine, going from a neutral to now negative charge. And here's the structure of the PAPS. And so this is required for cell adhesion, coagulation, also involved in G protein coupled receptors, extracellular matrix proteins, and so forth. Myristoylation or farnesylation allows proteins to then be bound to the lipid bilayer, so it attaches cytosolic proteins to the plasma membrane, and they're usually involved in signal transduction. The type of linkages for myristoylation can be an amide link or a thioester link. In the case of an amide link, myristic acid, which is 14 carbon chains, may be linked via an amide bond, shown here, to the amino group of the N-terminal glycine residue. In the thioester linked, you can see it's using a cysteine, and that cysteine links it as a thioester bond, shown here. Farnesylation is actually a thioether linked bond, and it uses C-terminal cysteine, and the C-terminal cysteine forms a thioether linkage with farnesyl or also geranyl groups. The cysteine is part of a carboxy terminal recognition sequence that's comprised of cysteine, alanine, and X, and so it gets attached to this cysteine, but then after it's attached, there's a processing of proteolytic cleavage that cleaves it, and so then you have this C-terminal cysteine shown here. Proline and lysine have also can be hydroxylated as a post-translational modification. Now, this is important in maturation and secretion in collagen. Occurs usually in the endoplasmic reticulum on procollagen. Requires oxygen, alpha-ketoglutarate, as well as ascorbate, vitamin C, and what will happen is you can have hydroxylation occurring on, in this case, 4-hydroxyproline. And then in the case of lysine, you have 2-oxoglutarate, oxygen, and ascorbate in the presence of iron, and you're seeing hydroxylysine being formed. So it's important for folding assembly of mature collagen and in terms of proline as well as a lysine hydroxylation. And you have different enzymes that can do the hydroxylation on the different sites of the carbon. So in this case, you have proline 4 hydroxylase, lysyl 5 hydroxylase, and prolyl 3 hydroxylase shown here. The gamma hydroxyproline stabilizes collagen triple helix, whereas the gamma hydroxylysine is necessary to form cross-links and attachment of glycosyl groups. Ubiquitin is prepared for conjugation to other proteins as a post-translational modification by ATP-dependent ubiquitin activating enzyme, or E1. So here is your ubiquitins. In the presence of ATP, you have this ubiquitin activation occurring with this activating enzyme. This is then transferred to E2, the ubiquitin conjugating enzyme, and then this forms a complex with E2 and E3 called the ubiquitin ligase, and then it allows for the tagging of the ubiquitin and polyubiquitin onto the protein, which gets targeted to the 26S proteasome for degradation, which chops the protein down and then also scavenges all the ubiquitin so that it starts a pool for further ubiquitination. So it requires ubiquitin ligase and the ubiquitin itself. The ubiquitin is about 76 amino acid. The terminal C-alpha group is covalently linked to other proteins through amide bonds through the alpha or the epsilon amino groups. And you can have activation of the E2, E3 complex either in the presence of ATP by phosphorylation or an allosteric transition caused by ligand binding, or allosteric transition caused by protein subunit addition. The identity of the N-terminal amino acid is important. So if the N-terminal amino acid is methionine, isoleucine, valine, glycine, proline, there's low degradation. If it's this RKH, EQDN, LFYW, AST, they undergo rapid degradation. So now there are also modifications in the N-terminus, and after that we'll talk about C-terminus modifications. Myristoylation occurs on the N-terminus. N-terminal methionine can be removed, addition of N-terminal residues to proteins, and end-ruled, and acetylation of N-terminus. In prokaryotes, initiating formal methionine is removed by deformylase enzyme. In prokaryotes and eukaryotes, initiating methionine is removed by ribosome-associated methionine aminopeptidases. Methionine aminopeptidases activity depend on the second amino acid, and it favors when the second amino acid is glycine, alanine, serine, cysteine, or threonine for removal. You can add amino acids to the N-terminus using tRNA protein transferases. The leucyl phenylalanine tRNA protein transferase attaches methionine, leucine, phenylalanine to N-terminal of proteins bearing arginine or lysine. Arginyl tRNA protein transferases attach arginines to proteins with N-terminal glutamic acid or aspartic acid, and they're implicated in tagging protein degradation. NN rule for protein recycling. So this NN rule pathway is part of the ubiquitin system, and we've talked about ubiquitination. NN rule pathway degrades cytoplasmic proteins on the basis of their N-terminal residues. Distinct versions of the NN rule have been identified in E. coli, Saccharomyces cerevisiae, and mammalian cells, suggesting that cellular turnover of cytoplasmic proteins bearing certain N-terminal residue is broadly conserved. Acetylation of N-terminus. So 50 to 90% of eukaryotic proteins synthesized in cytoplasm bear acetylated N-termini, and alpha acetyltransferases catalyze the acetylation with acetyl coenzyme A and acts on 20 to 50 amino acids off the ribosome. And so, like histone acetyltransferases, this then, instead of acetylating lysines, what it does is acetylates the N-alpha amines. Acetylation occurs whether or not initiating methionine's presence, but it prefers, GAST, prefers methionine when methionine is followed by DE and N. Now, modifications of the C-terminus. So you can have C-alpha attachment of glycosyl phosphatide and inositol, Farnesyl and Geranylgeranyl attachment to the C-terminus of cysteine, amidation of the C-terminus, and then reversible removal of C-terminus tyrosine. So in terms of a C-alpha attachment of glycosyl phosphatide and inositol, this occurs at the terminal alpha carboxy group. It's comprised of an ethanolamine linker via an amide bond, glycan, and inositol phosphate, shown here. Advantage to simple membrane proteins is that in that this type of anchoring may be regulated by enzymes that hydrolyzes the linkages. Farnesyl and Geranylgeranyl attachment. Like I said, they're attached to sequences that have cis AAX, where A is equal to leucine, isoleucine, or valine, and X is any amino acid, and then afterwards it gets processed or cut off, and we had shown this before. Amidation of C-terminus. They're found in peptide hormones. They contribute to stability. Amide group is derived from the C-terminal glycine after proteolysis. So as you can see, you have proteolysis occurring, and then that's how amidation occurs in this in the sequence. And this process usually occurs in secretion granules. The C-terminus of alpha chain tubulin is subject to reversible incorporation of tyrosine by tubulin tyrosine ligase, and the removal by tubulin carboxypeptidase. So microtubules rich in either tyrosinated or detyrosinated tubulin can coexist in the cell. This is ATP dependent, this process. Tubulin here exists as a soluble heterodimer alpha-beta tubulins. They undergo assembly, forming filamentous microtubules over here by first forming a protofilament, and by the enzyme tubulin tyrosine kinase forming these microtubules. This is important for mitosis as well as motility and transport. So in summary, I've discussed post-translational modifications. They're very necessary for protein function. They allow for correct protein folding, organism development, cellular signaling, motor proteins, regulating degradation, and so much more. And so in this chapter, you'll start, or in this lecture, and the papers that you'll read, you'll start to learn about them and the different types of post-translational modifications and how people study them. And it is my expectation that you know all of them and what residues are involved in which types of post-translational modifications.